Okay, here we are back for our fifth lecture on Chapter 20, Enzymes and Vitamins. And believe it or not, we're still not going to discuss the vitamins very much. So obviously you know what Chapter 20, Lecture 6 will be about. Our particular focus for Lecture 5 here is the regulation of enzyme activity. So we've talked a lot about enzymes with their basics, their naming. Most recently we talked about inhibitors and how the inhibition of enzyme activity occurs. So now we're ready to look at the regulation of enzyme activity. Now, zymogens or proenzymes are inactive forms of enzymes, and they're activated when one or more peptides are removed. So this is very good because obviously if the enzymes are so good at catalyzing these reactions, then we've got to have a way to transport them where we need them without them necessarily acting in an area that we don't want them to act. So these zymogens, these proenzymes, they're inactive. They don't do the enzyme's function. So therefore, they can be transported to the part of the body where the active form is needed without doing that active enzyme activity along the way. So this is very ingenious, the way that life evolved to have this very complex chemistry that is there where you need it and not where you don't. It's amazing, really super stuff. One example of these proenzymes would be the digestive enzymes, right? We don't want them destroying proteins all along the way to that desired activity. So instead, they're produced as zymogens in one organ and then transported to another, like the pancreas, where they are needed. The way you activate these digestive enzymes is by removing small peptide sections. So we see in table 20.7 some examples of the zymogens and then their active forms. So we have proinsulin is the zymogen, then it's produced in the pancreas, it's activated in the pancreas as well as insulin, the active form of that enzyme. Chymotrypsinogen, these are a little more difficult to say sometimes, especially when they get quite that long. Another one that's produced in the pancreas, however, this one acts in the small intestine, and its active enzyme is chymotrypsin. So obviously that ogen ending means it's going to be producing chymotrypsin once it's activated. Pepsinogen, we see that same sort of ending. It's produced in chief cells and then activated in the stomach as pepsin. Trypsinogen, produced in the pancreas, activated in the small intestine as trypsin. Fibrinogen in the blood and then activated in damaged tissues as fibrin. And then prothrombin, also produced in the blood, also activated in damaged tissues to form your thrombin, right? We don't want the fibrin and the thrombin active, or they can cause trouble in healthy tissues, but once there's damage and we need to stop bleeding, for instance, then you're definitely going to want the active forms of those particular enzymes. Okay, so here's our cartoon showing the activation of trypsinogen and chymotrypsinogen. So we've got our chymocitrininogen and our trypsinogen just floating around there, and then eventually we get the spontaneous removal of the group that causes the trypsinogen to form trypsin, and then that trypsin can help coerce the chymotrypsinogen to lose its portion there and become the active form chymotrypsin, and the trypsinogen can also be acted on by trypsin to form more active trypsin. So we get these cascades, as they're known, and it's really, really elegant chemistry that happens in the body. So we've already mentioned that sometimes we want these enzymes and sometimes we don't, and sometimes we want them in a particular place and not in another, and that's where those proenzymes come in. Let's talk about a feedback control. So these allosteric enzymes, they help the enzyme in a reaction sequence that binds a regulator molecule that's different than the substrate. So we can have a positive regulator, and that's when the allosteric enzyme enhances the binding of substrate and accelerates the rate of reaction. And we can have a negative regulator when it prevents the binding of the substrate to the active site and slows down the rate of reaction. So we need to be able to both transport inactive forms, as we've already talked about, and be able to speed up or slow down reaction rates by having these allosteric enzyme regulators. So one example of the enzyme regulators would be a feedback control. So in feedback control, a product acts as a regulator. So an end product of a series of reactions binds with that first enzyme in a sequence that when sufficient product is present, it shuts down the reaction. That's a really nice way to do things, right? If your enzyme is there to create product because you need it, well, once your enzyme is creating so much product that there's excess product around, that end product can go back and be an allosteric inhibitor. It can bind to the non-active site, right, as we show in our cartoon there, and prevent more substrate from binding, alter that active site so that substrate can't bind. And then if we do this reversibly, then if the end product levels drop to the point where they're coming off of that allosteric site, then the enzyme's active again and it can make more product, and it's just really a neat biological equivalent of the old thermostats, right, which would be an example of a feedback control in a non-biological system. Okay, so we had our cartoon at the bottom of the previous slide for a generic example. Here, let's talk about a specific example, feedback control by threonine deaminase. So when isoleucine accumulates in a cell, it binds to threonine deaminase, the first enzyme in the pathway from threonine to isoleucine. So that results in a change in the shape of the threonine deaminase, which blocks the binding of additional threonine, and the feedback control decreases the synthesis of isoleucine, which obviously if we're getting a buildup of isoleucine, it's not so critical that we're synthesizing more of it by that threonine deaminase enzyme. So there we have it. Threonine converts in a series. Here it's a 5 enzyme long series to get to isoleucine, but that isoleucine at the end, once it accumulates in sufficient quantities, it can go back and bind to that first enzyme, the threonine deaminase, and stop this whole process, at least until the isoleucine level drops to the point where maybe we need that threonine deaminase reactivated in order to make more. Okay, so I know everyone's eager to learn about the vitamins that we've kept you from for so long, and the next lecture is the last lecture in the chapter, so you will be learning about vitamins there. But first we'll end this chapter with a learning check, as we have done for all the lectures in chapter 20. In this particular learning check, you're asked to identify each statement as zymogen with a capital Z, allosteric enzyme with an A positive regulator, PR, or feedback control, FC. So read those descriptions for A, B, C, D, and decide which best describes that statement. Pause the video here while you make those assignments and start us back up when you're ready to check your work. Good luck. Okay, hopefully these are the same solutions that you arrived at. So for A, an enzyme in a pathway that controls the rate of the reaction, that's an example of A, an allosteric enzyme. For B, it speeds up a reaction by combining with an enzyme in the pathway. That's going to be an example of a positive regulator. So it's speeding things up by combining with that enzyme along the path. For C, the removal of a peptide activates the enzyme, so that we must have a proenzyme or an inactive form of the enzyme known as a zymogen, Z. And finally, D, some product binds to the first enzyme to limit the synthesis of additional product. That's an example of our feedback control, just like the isoleucine we talked about just before our learning check. So there we have it, the 4 different descriptions and the identity of each of those descriptions. So if you are all good with these and you arrived at the same results, so wonderful. If that's not the case, hopefully it makes more sense now, and if it's still being a source of frustration, then please reach out for some help. Otherwise, we'll see you for the next and last lecture in chapter 20, where I promise we will talk about vitamins.